Somebody please confirm you can hear me.
Yes.
Great. All right, my computer is going to be slow. I think you can see my sheet now. The lecture is about 81% done, so it's going to be slow until this finishes. So just bear with me. All right. Are there any questions before we begin? Not hearing anything, so let's go on. Today is January 16th, Thursday. And in the lab, we're supposed to be discussing lab 2 on aseptic transfers. Let me remind you that you should be reading in the textbook in chapter 3 through the end of the section covering compound light microscopy. You need to have that read before beginning lab 3 and lab 4, which we will do next week. Any question about any of that? Let me also remind you that the plagiarism quiz, which was due actually last Saturday, you can finish it for 100 points, 100%, I guess, for by this Saturday. So if you're not at 100%, please get it up to 100%. If you do not get 100% on the plagiarism quiz, you'll get 0 points on the 100-point infective disease project. Any question about any of that? All right, let me remind you also that this lab 2, the worksheet is due this Saturday. And if you do not get 100% on the first grading of lab 2, you will be allowed to turn it in again for a second time, and the total possible points will be up to 100%. So lab 2, you get to turn this lab in 2 times if you don't get 100%. You will turn it in obviously this Saturday. And if I get it graded to you by Tuesday, then the second grading will be due not a week from this Saturday. So a week from this Saturday, whichever day that would be. And good, the lecture Zoom page just finalized. My computer will now work a little faster. Let me see if I can call up the calendar. So this Saturday is the 18th. So if I get it graded to you by um, at nighttime on the 21st, and that could be early on the 22nd, meaning very early on the 22nd, it would be due the 25th. If for some reason I do not get it graded by early on the 22nd, I will then postpone the turn in date until the 1st. But there's only one lab, so I shouldn't have any trouble getting it graded on time. Any question about that? That differs for the labs in week 1, where lab 00 and lab 01, you only get to turn them in one time. Now, if you like one of the turning in the labs one better than the other, there's something you can do about it, and that is come into the student discussions and give a comment on whether you want the lab to be turned in one time or 2 times. Normally I did it um, 2 times, but last term there were at least 2 students who complained about turning in the labs 2 times. They also complained about my reading the lab and grading each question. And yes, I will read all of your answers, and you must submit correct answers for getting full credit. Generally, all you have to do is look up the answer in the lab module. There's a few questions you have to do a search online, but I've always got that labeled when that's the case. Almost all of the questions you can get from just reading the lab module. Anyways, if the majority of the students request to turn it in one time, I'll do that for the future labs. If the majority of students request to turn in the labs 2 times, then we will do it that way. So you guys get to decide. And you'll get an extra credit point if you give an answer. When is that? I think it's this Saturday. I don't remember. Yeah. Whoops. March 15th. It's already closed. Let me look into that. That probably should have been March 16th. Oh, wait a minute. March 15th. That's not right. All right. I understand. No, the extra credit. There it is. It's due January 18th. So for the extra credit, you have to turn it in by, I think that's Saturday. Yes, at 11:59 p.m. Any question about any of that? All right. If there's no questions about anything, let's begin with the lab. Lab Module 2, Aseptic Transfers. Upon successful completion of this lab, you should be able to understand the purpose of using aseptic transfer techniques. You should be able to determine if aseptic transfer is used from the data provided, meaning everything we do in this lab will be done online, so you're not really going to be able to do aseptic transfer. And then be able to define the following terms. What does ubiquitous mean? What is aseptic transfer? What is inoculation? And what is a contaminant? In this lab, there will be some video to watch. Any questions about anything? All right. In our local environment, microorganisms are ubiquitous, meaning you can find them essentially everywhere. This is problematic when you're working in a microbiology lab because we generally want to work with a pure culture. A pure culture is one that contains only one species of microbe, or only one species, period. In the mid 1800s, you don't need to know that, but you should know Louis Pasteur developed the technique for making aseptic transfers to minimize the risk of contamination of the cultures. He also came up with a way of obtaining a pure culture, and we'll talk a little bit about that later. His way of getting a pure culture was actually difficult, and that is he did a liquid culture, meaning a broth. He would make different dilutions of his solution containing a bacteria and or microbes, and he would take that dilution and then make 10 replicas, meaning have 10 test tubes of that dilution. Then he would take another dilution, meaning 1/10 of that dilution, and make 10 replicas of it, and then uh, uh, grow those up, and then take that dilution and make a dilution of that, 1/10 of that, etc. And the point is, he had, well, certainly close to 50 to 100 tubes of um, his dilution. Sometimes he would have hundreds of tubes. And what he looked for would be one dilution where only one tube out of 10 of that dilution had growth in it. And he correctly concluded that if you got growth in only one tube and no growth in 9 tubes, that the tube having one growth was likely started from one or very few cells, which meant there was a high probability that that growth was a pure culture. And that's how he got his pure cultures. It was a difficult technique, and Dr. Koch came up with an easier technique, which we'll talk about, I think, in a future lab. Maybe it's this lab, but I think it's a future lab, where he streaked out for getting isolated colonies on a solid media, solid agar plate. And then he took the cells from a isolated colony, streaked it out a second time on a second plate for isolated colonies. And if you take cells from A colony that's gone through 2 colony isolation events, the chance of getting a pure culture is quite high. Over 95% of them would be a pure culture, and that's good enough for use in microbiology, meaning you didn't quite get 100% chance, but it was very high. It was close to 100%. And you never really get a 100% chance, but it's quite high. In this lab, we'll learn about contamination, and that's accidental inoculation of other microbes to our culture, as well as we'll learn how to make aseptic transfers. Any question about any of this lab so far? All right. So when you're working in a microbiology lab, you never lay the test tube cap or the petri dish lid down on the table. Let me see if I can show you this. So we're going to pretend this is a petri dish. Hopefully you can see that. Let me stop my sharing. I'm not seeing it. Let's go here. There we go. Still don't see it. Boy, I'm still not seeing it. Ah! Can you guys see this? Oh, there it is now. I'm not seeing the color yellow, but there's my finger right there. So there it is. So you never lay the lid down on the counter, either that way, because it can pick up microbes here, and or put it there because microbes can fall into the lid from the air. And that's true for a cap as well. I'm having trouble starting the share again. Why is this? There it is. Not sharing me. So what you do with the cap is you hold it in one hand by these fingers, and then you hold the loop with the same hand, but with those fingers and the thumb. And with the lid, you hold it with the other hand, and then you hold the loop with the other hand. And when you're using the loop, let's pretend this is the loop. I don't know if you can see that or not, hopefully. And what you do is you keep the lid partially over the container. Obviously, the container has to be open to get the loop inside. And so something falling right here can get into your container and contaminate it. But most of the things will fall here, in which case they do not contaminate the petri dish. And then you use the loop, streaking it out on the plate, where you have the lid partially over the container. And that was discussed last time. It might not have been shown in the lab, but that was the correct answer for one of the lab questions. And here also, it's shown, I haven't talked about it, so let me talk about it. Let's go this way. Hopefully you can see this dish, which we're going to pretend is a test tube, a real big test tube here. And what you do is you hold the lid with your pinky and maybe the other finger in your palm of your hand, and then come into the tube. You don't have the tube set directly up because anything falling down can fall into the tube. What you do is you hold the tube at an angle, steep as you can. If it's solid, you can have it even sideways where there's very little chance it's going to fall in. But if it's liquid, you have to have it bent at an angle. And the idea is you decrease the surface area that something can fall and then contaminate the tube, because most of the things will fall here, which will not contaminate the tube. And then you inoculate the tube with your loop and then quickly cover the tube to prevent things from falling into it from the air. Any question about any of that? Quickly, leaving the tube open and the lid partially open, or the petri dish partially open, for as little time as possible. And that will reduce airborne contamination, reducing it to a level which is acceptable in microbiology lab. Meaning occasionally you'll get something that happens to fall in, but most of the time you will not. Any question about that? And that's good enough in a microbiology lab. Some people said work in an airflow hood, which is sterile airflow, and therefore nothing can fall into the tube. We don't have a device like that in the microbiology lab, and I know of no microbiology lab that has them. So all the microbiology labs, at least the ones I know in the country, do not have them. We simply work this way, and it's good enough for microbiology. So when you're working, work efficiently, do not talk. When you're transferring and have something open, sterilize the loops and needles before and after they're used. Making aseptic transfer. In order to inoculate the cells into a new sterile media, you must make sure that everything is sterile before you work with it, and that everything that can potentially contaminate the sterile media is kept out as best as you can. And this is showing you some of the tools: sterile media and a sterile tube, which obviously a test tube rack for holding them, either a Bunsen burner or a back-to incinerator. We in the microbiology lab no longer use Bunsen burners. The only exception would be, I think once we had a lab. It was in the daytime, and so light came in from the windows. And we turned on the Bunsen burners because the back to incinerator did not work. But other than that, we always only use back to incinerators, which becomes the stone inside becomes red hot. You put the wire loop inside until the metal portion of the wire loop, or the loop portion of the wire loop, whatever you want to call it, turns red hot. That will sterilize the loop. You then need to cool it, and then pick up your cells from the stock culture, and then stock culture, and then transfer it into your sterile media. And that's really aseptic transfer. Any question about any of that? All right, you should know the terms in this lab. You're not physically going to be performing any of these activities, but you will be watching videos of the activities, as well as we'll discuss the activities, or you'll have to read the activities, and you should know and understand the material in the video and in this lab. For procedure 1, we're going to transfer sterile liquid water using a glass serological pipette, transferring the water into a tube containing media. And we have a little video for both procedures 1 and 2 for you to watch, discussing or demonstrating proper aseptic transfer techniques. As I stated, in procedure 1, you're going to transfer 1 ml of sterile water and put it into a tube of nutrient broth. When you're transferring water in a glass pipette or a glass pipette, or even a plastic pipette, the water will form a meniscus. It will not form evenly. It'll be especially high in meniscus if it's glass, but it will form a meniscus also if it is plastic. Where do you read the meniscus? In science, we read the meniscus at the bottom of the meniscus. We do not read it at the top of the meniscus. We do not read it in the middle of the meniscus. Any question about that? So that's adding 1 ml of water, assuming we have 1 ml there, which I guess is what that's trying to show. And then you put the mill of water into the 9 mils of broth. You can read about the procedure here. And then you incubate it overnight at 35 degrees Celsius in the incubator. And then come down here this is showing you the broth culture tube before you have opened it and put cells in it. Broth cultures are a problem because it's difficult to determine if contamination has occurred, particularly if the contamination is less in number than the cells that you add to the tube. And that's because all the cells will mix together, so you do not see any colonies. You do not see one colony of one color and another colony of different color. And so contamination is much more difficult to determine when you're growing things in a liquid medium. Any question about any of that?
Just real quick to clarify, so the only way to thoroughly tell if you have a pure culture is to streak for isolation.
That's true
Okay.
for a liquid culture, at least. For a solid culture, you don't need to streak for isolation. We'll talk about that in a little bit. For procedure 4, we'll talk about it. All right, remember you always label things on a piece of tape with your name, the date, the type of medium, and the inoculum, whatever you're growing. You may write, this is optional, the incubation temperature. Then come down to this table for determining the results after you grow the tube overnight in the incubator at 30 or 35 degrees. I think it's 35 degrees for getting the results and getting your conclusions. For procedure 1, if the nutrient broth is clear, no organisms are growing in the broth, and aseptic transfer was successful. However, for procedure 1, if the nutrient broth is turbid, is hazy or cloudy, then organisms are growing in the broth and aseptic transfer was unsuccessful. Any question about that part of the table? All right, there is procedure 1, tube for procedure 1. Come down for the question. So this is the results that we're providing for you. You need to answer question 0. Describe the initial clarity of the nutrient broth prior to inoculating it, meaning prior to adding the sterile water or the paper disc for procedure 2. And that's shown right here. So is it clear or turbid? Right there. And for question 1, covering procedure 1, describe the clarity of the nutrient broth after inoculating with sterile water and overnight incubation at 35 degrees C. Was it clear or was it turbid? For 1B, based on these results, what conclusion can be drawn? Your conclusions must use wording similar to the table above. Coming to the table. Where did I miss it? There it is. So if the nutrient broth is clear, no organisms are growing in the broth, aseptic transfer was successful. If, on the other hand, the nutrient broth is turbid, then organisms are growing in the broth, and aseptic transfer was unsuccessful. You must, to get full credit, tell me whether there's growth in the tube and whether aseptic transfer was successful or not. You do not need to use exact wording, but if you use exact wording, that's okay. And if you do quote from the lab, you do not have to put it, quote from the table. I mean, you do not have to put it in quotes and say you got it from the table because it'll be quite apparent to me. Okay. But you may do that. Like I said, you don't need to do that. And if you use your own wording, your conclusion, just use wording similar to the table above. Otherwise, you won't get full credit. And then 1C lists 3 sources of error with a septic transfer technique that could explain why nutrient broth showed growth of microbes in the tube. And just fill in each asterisk. Any question about procedure 1? If not, let's go to procedure 2. Procedure 2, you're going to be transferring a sterile paper disc using forceps. And we have a little video for you to watch on uh, watching proper aseptic transfer. I think I forgot to mention that last time. So watch that video and for procedure 2, you're going to use a septic transfer technique to transfer a sterile paper disc using clean forceps. What we do, I don't think I have forceps here, so we'll pretend my fingers are forceps. We have a disc for the paper, a dish with paper disc in it. Come in with your forceps, pick up a paper disc that's sterile, and you need to sterilize the forceps by putting them in alcohol and then bringing the alcohol next to the back to incinerator so that the alcohol lights. It'll actually be hot enough to light the alcohol. And then you don't need to cool the forceps because we won't be touching anything alive. But then go pick up the sterile paper disc and put it in the tube of broth. And then incubate the tube overnight at 35 degrees Celsius in the incubator. So there's the tube we're adding the paper disc to. And then use the results you see in the tube from the table for procedure 2. If the nutrient broth is clear, the conclusion is no organisms growing in the broth and aseptic transfer was successful. If, on the other hand, the nutrient broth is turbid, then organisms are growing in the broth and aseptic transfer was unsuccessful. Come down to here, there's the picture of the tube after incubation for procedure 2. And there you can actually see the paper disc on the bottom of the tube. And then come down to the questions. Question 2, the results of procedure 2, P2. Describe the clarity of the nutrient broth after inoculating with sterile paper, the sterile paper disc, and overnight incubation at 35 degrees C. Was the tube clear or turbid? And based on these results, 2B, what conclusions can be drawn? Your conclusions must use wording similar to the table above. Any question about any of that? All right, let's go to procedure 3. Procedure 3, you're going to transfer Serratia marcescens to a broth. Remember, a broth is a liquid culture. Serratia marcescens is something that grows in a broth that looks pink, and so that will help you determine whether you have Serratia marcescens growing in the broth and whether you have contaminant or not. Now remember, it's a liquid culture, so even if the broth does appear pink, you do not know for a fact that there is not minor contaminants in the broth because the minor contaminants will be overshadowed by the major growth, which would be, say, Serratia marcescens. For procedures 3 and 4, we have another video for you to watch how to correctly transfer from the bacteria stock culture to a sterile broth culture and to a sterile agar slant, and that'll be procedure 4. So you can watch that video. You only need to watch this video into 4 minutes and 23 seconds of the video. After that, you do not need to watch the video any further. And there's actually an error the instructor who made the video made. The error is around 3 minutes and 5 seconds to 3 minutes and 20 seconds, in that range. And you can actually hear sizzling when he puts in the loop into the stock culture of E. coli. And that's part of the error. The question is, can you tell me what the error is? What was the error the instructor made? You can write about it. I'm not going to give you any credit about that, but you should try and figure out what the error was. Okay, any question about that? All right, procedure 3, you're going to use aseptic transfer technique to transfer Serratia marsens to the sterile broth. Serratia marsens grows pink, but remember broth cultures are problematic because it's difficult to determine if contamination has occurred because all the cells mix together, particularly if the contaminant is lower in number than what you introduced in, meaning if the contaminant was only 10% and 90% was Serratia marsens, the color will be pink and you will not see the contaminant. Remember with the tube, you should always label them with your name, the date, the type of medium, the inoculum, in this case, Serratia marsens, and then the temperature you grow it at can be optional. Interpretation of results for procedure 3. If the nutrient broth is clear, then no organisms growing in the broth and aseptic transfer was unsuccessful. If, on the other hand, in procedure 3, the nutrient broth is turbid, hazy or cloudy, then organisms are growing in the broth and aseptic transfer was successful. Why in procedure 3, if we have organisms growing in the broth, aseptic transfer was successful, but for procedure 1, if no organisms are growing in the broth, aseptic transfer was successful. So why is that changed? Anyone? Or what are we doing in procedure 1? Procedure 1, we're adding 1 ml of sterile water to our broth. What are we doing in procedure 3?
We're trying to inoculate a broth with a culture.
Yeah, you're trying to inoculate your tube of broth with Serratia marsens. So we expect growth in procedure 3 because you're inoculating it, you're putting cells in the tube. So that's why aseptic transfer is successful if you get growth with procedure 3. But if you get growth with procedure 1, aseptic transfer was unsuccessful because we put sterile water and nothing else in the tube. All right, then come down to the picture. There is the picture of procedure 3 after we inoculated the tube, then incubated it overnight. Pull it out and take a look at it. And then come down to the questions, answer the questions. For question 3, the results of P3 describe the clarity of the nutrient broth after inoculating with Serratia marsens and overnight incubation at 30 degrees C. Is it clear or is it turbid? We're talking about the tube. Based on these results, meaning 3B, what conclusion can be drawn? Remember, your conclusions must use wording similar to the table above. For 3C, how do you know whether you're transferred the correct microbe, Serratia marsens? Anyone? How would you know if you transferred Serratia marsens and got it growing in the tube?
So we're not gonna know 100% for certain, but there should be a growth, a pink growth that we're trying to look for.
Yeah, and this question is not answering is there contamination, but if the growth is pink, you can assume that you transferred Serratia marsens into the tube, and that's giving the pink color. But if there's contaminant, and it's a major contaminant, meaning more of that than the Serratia marsens, then the color may change, in which case you know you have the contaminant. But you're right, and that is if there's minor contaminants, you wouldn't know about the minor contaminants because they will be overshadowed by the Serratia marcescens, and the tube will continue being pink. And that's just your answer. All right, for procedure 4, we're going to be transferring Serratia marcescens onto an auger slant. And there's a little video for procedure 3 and 4. I think I already talked about that. You only need to watch the video to 423. You only need to watch the video one time, by the way. But it's for both procedures 3 and 4. Procedure 4, you're going to be using aseptic technique to transfer Serratia marcescens to a sterile auger slant. When Serratia marcescens grows on an auger slant, it will be red in color. So it will be pink in a liquid broth, but when growing on a solid media, Serratia marcescens actually appears reddish. Contamination can be more easily be detected on a slant than in a broth. What you do is you pick up on your, where's my pencil? On your loop, the stock culture, and then streak it on your slant in an S-type motion, going like that. So you inoculate the slant. And if you see only red in the slant, there's no contamination you can see. But if on the other hand, there's one area or one colony area that is not red, then you know you have a contaminant because Serratia marcescens should grow red. Any question about that? So if there's an area of the streaking that's not red, then you know you have a contaminant. All right, for procedure 4, if the slant does not show confluent or colony growth, then no organisms are growing on the slant and aseptic transfer was unsuccessful. If the slant does show confluent or colony growth, organisms are growing on the slant and aseptic transfer was successful. Here we have the tube, the slant for procedure 4. And as you can see, we have so much growth that we don't even see the streak. This entire region is growing, the Serratia marcescens. And then there was a drop of water on the tube, and so the Serratia marcescens swims, and so it actually colonized this entire area as well, because we only streaked right here. But it swam in the water and then spread to there. That's probably why it spread this way, grew outside the slant, and there's so much growth and Serratia marcescens moves that it's growing in all this region there. And if you look very closely, you can see there's a little bit of media there, a very tiny bit of media there, where there's no Serratia marcescens growing. And that's just because we didn't streak all the way across the slant. For some reason, the person only streaked right there. Didn't streak all the way across.
Odd curiosity question.
Uh-huh.
Um, are most forms of Serratia, are they pink in color or is it just Marquesans?
I didn't quite understand the question. Maybe I misheard or didn't hear something. What did you say again?
Are most species of Serratia, when we see them in real life, are most of them pink in cultures or in tests, or is it just Marquesans that's pink or red when it's grown?
Only Serratia marcescens is
Okay.
pink or red when it grows. Most bacteria species will grow either white or off-white, okay, or beige or something like that, off-white. Uh, there are a few that have color. Obviously, Serratia marcescens, when you grow it on a solid culture, it's reddish. In a broth, it's pink. And there's a few others which are colored. Micrococcus luteus is yellow. Serratia, star is not Serratia. Staphylococcus aureus is yellow. But most of the species we work with will be unpigmented and they're either white or off-white or some color like that. All right, then go to the questions for uh, procedure 4. Question 4, is there bacterial growth on the slant? Just answer yes or no. How do you know? So you need to explain how you know, just simply. And then 4C, does it appear to be the bacterium you inoculated or contaminant? And then explain why. Okay? Any question about any of the questions? Any question about any of this lab? All right, if there's no questions, I will be here until 8 o'clock, or a student logs out, whichever comes first. And I'm here to help you guys with any questions in the lab.
Be over like,
I didn't hear you. You stopped. Your microphone stopped.
oh, sorry. Can you still hear me now?
Yeah, I can hear you now.
Okay. So quick question for our quiz that's next week on Wednesday. Is it basically going to be over like as far as we got up to in lecture and lab today?
Let me check the syllabus. I don't know the answer to that. So next quiz will be quiz 1. It will
Mhm.
cover labs 0, 1, and 2. So yes, it does cover the lab today.
Okay.
And then it will cover chapters 1 and 10, and only the start of chapter 2.
Okay.
We're further than just the start of chapter 2.
Okay.
Okay?
Sounds. Thank
All
you
right.
so much.